Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Welcome everybody back to In the Interim. I'm your host, Scott Barry, and I'm joined by a couple investigators from down under. We are going to talk about the SNAP platform trial. So let me first introduce our guests for today. I have Professor Stephen Tong, and he's a practicing infectious disease physician at Royal Melbourne Hospital and a professor of infectious diseases and clinical trialists at the Doherty Institute at the University of Melbourne. And one of my more favorite titles, he's the head of adaptive platform trials. That's really cool. At Melbourne Center for Clinical Trials. He co-leads the Staphylococcus aureus network adaptive platform trial, and I hope that's the last time I have to say that, and which is recruiting participants, 150 hospitals, 13 countries, and we're to get into the SNAP trial as this is the topic of today's discussion. And he is joined by Professor Josh Davis, who's also an infectious disease and general internal medicine physician at the John Hunter Hospital in Newcastle, New South Wales, Australia. He's also a professor of infectious diseases at the University of Newcastle and director of the infection research program at the Hunter Medical Research Institute. And he co-leads the SNAP trial with Professor Tong, as well as the Roadmap trial. We probably won't talk about Roadmap, but this is an adaptive platform trial in management of hip and knee infections. And interestingly, Josh and Steve did their PhD studies together in Darwin at the Menzies School of Health Research in Darwin, and have had a productive ongoing relationship that continues to grow for the last 15 years. So Josh and Stephen, welcome to In the Interim.
Thanks for having us.
Yeah, I enjoyed listening to In the Interim, so it's a great honor to actually be invited to come and talk on it.
Wonderful. It's exciting. So first, I want to ask about Darwin. Darwin is such an interesting place. I've never been there. But it strikes me as a very isolated area, kind of a unique area. Seems like a really neat area to do work in infectious diseases. Is your work and time in Darwin part of this story?
Yeah, it is. I mean, that's where we met each other and started collaborating, but also where we planned and ran the Camera 1 trial, which was the first trial we did together in MRSA bacteremia, Staph aureus bloodstream infection, that then led on to a series of steps that ended up with the SNAP trial. We were both there because it's a place that infectious diseases trainees often go to spend a few years in Australia because it's fantastic infectious diseases training environment and research environment as well up there.
So it's part of the tropics, and so there's lots of tropical diseases. There's an environmental organism called melioidosis, which you only see up in northern Australia, at least in Australia. There's lots of dengue, malaria that comes through from Southeast Asia. So really great place for infectious diseases training. And also First Nations health is super important there as well. So there's a large Aboriginal population. So a lot of us go there to learn about, I guess, the disparities that we see in our country.
Now, we're going to get to the SNAP trial, but do you have a site in Darwin?
We do.
Okay.
Absolutely. So,
okay.
And as Josh said, that's where it all started. We started studying Staph aureus because we saw a high burden of disease in the Indigenous population in the Northern Territory. In fact, my PhD was done up in Darwin, specifically on Staph aureus in Northern Australia.
Okay, so that comes to the SNAP trial. And the SNAP trial, it says the aims are to improve treatment outcomes for patients with Staphylococcus aureus bloodstream infections. So tell me how this trial came about.
I'll go.
Yep.
So Staph aureus is a major burden of disease globally. Of the 33 top bacterial pathogens that cause disease, it's actually number one. So outside of tuberculosis, of the top 33, it's number one. It's the only one that's estimated to cause more than 1 million deaths per year, of which about 300,000 are due to bloodstream infections. So this is the most serious form of Staph aureus infections. Staph aureus causes, skin infections, infections of other parts of the body, but once it gets into the bloodstream, that's when it's most serious. It causes a mortality rate of 20 to 25% per episode. So it's a big deal, and so it's important to study. As infectious diseases clinicians, we see lots of this. So every week in our hospital, we're on clinical service. We're seeing 2 or 3 patients per week with this condition, and so it's one that really needs study to try and improve outcomes. That mortality rate of 25% or so per episode has been stuck at that for probably the last 30 to 40 years, I think, and we haven't seen improvement in our therapeutics for staph bacteremia.
So what? So you're talking to a statistician here, but it may help. What does it mean to have a bloodstream infection? And I suppose it's moved from an organ, and the blood itself is infected. What does that mean, Josh?
Yeah. Well, I guess technically it means we have found it. We've detected it in the blood using blood cultures. But generally, it starts from the skin, from either a skin infection or through a cut or a scratch on the skin, gets into the bloodstream. And it being in the bloodstream by itself isn't the primary problem. It's that the bloodstream is the route of transport to everywhere in the body. And then so the Staph aureus germ can then spread to other organs, and often does do so, and it ends up infecting bone, heart, liver, etc. So bloodstream infection means the bacteria, rather than just sitting on the skin, has invaded into the body.
Okay. So I could have an infection, but it is, it's on the skin, it's outside, it has not gotten inside in it. Okay. So it's quite the impact. And just this is a U.S. thing, that people in the U.S. are going to have this infection. This is a first world problem. This isn't just a Darwin problem.
No,
it's everywhere. It's very prevalent in the US. It's very prevalent and probably less well described in low, middle income country settings, but I'm sure it's prevalent there. It's just there's so many other causes of infections and burden of disease that it doesn't quite, have the prominence that we sometimes see in high income countries. It's an organism. It's a really interesting organism in that once it does get in the bloodstream, it has a whole variety of virulence factors. That means it can escape the immune system. It can form what we call biofilms. So if there's any foreign bodies like pacemakers, heart valves, prosthetic heart valves, prosthetic joints, that it gets really sticky and it sticks to those. And so it becomes very difficult to treat. And it's not just antibiotics, therefore. You need to remove infected devices as well. And in high-income countries, as more and more people get joints, replaced, get prosthetic valves, it becomes more and more of a problem.
Okay. So clearly a high unmet need, 1 million deaths a year, you said, or 1 million deaths a year, and things have not moved in decades. So what was the genesis? I mean, presumably the trial was to change that. The genesis of then the SNAP trial to investigate this.
So we can follow on from that story that Josh started. He and I were seeing a lot of patients with MRSA, so the more resistant from methicillin-resistant Staph aureus bloodstream infections in Darwin. And we actually just sat at the table and said, what are we going to do about this? And so we started a small pilot trial with colleagues in Australia that got to a 60-patient randomized clinical trial. That proved some feasibility for a larger study of about 350 patients with MRSA bloodstream infections. That one was called CAMRI 2, and now we were involving 4 countries. And once we finished that trial, we started talking to Steve Webb, who many of your listeners might know. He heads up REACT in Australia. And Steve started talking to us about adaptive platform trials. And we just thought, well, we've got these two things coming together, staph bacteremia. We've got some runs on the board with a couple of studies now. We'd like to apply an adaptive platform trial to this particular disease. And that's really where it started. So those discussions with Steve and Josh and myself, we wrote a couple of grants, and the second time we wrote that grant, it got up, and we therefore started the trial.
I guess maybe to add to that MRSA camera trial was about combination antibiotic therapy, so using 2 antibiotics instead of the standard 1 to treat MRSA bloodstream infection. And the next step for that would be that we wanted to do was to do a larger trial of many more patients of the same intervention. But in parallel, there are a whole lot of other questions in management of this infection we were interested in, and that there had been preliminary data either generated by us or other groups that we wanted to turn into trials. So we could have done that in serial, which would have taken 50 years, or we could have done it all at the same time, which is kind of what SNAP is.
Yeah, so we'll get to that. This is a very cool factorial trial where you're doing multiple investigations on the same patients. But I think it seems like part of this story, though, is the culture of staph or bacterial infection research. Is there a deep history of clinical trials in this area? What's the background in this area?
Yeah, that's a great question, Scott. And I think in infectious diseases are somewhat well behind lots of other craft specialties. So, we at the cardiology trials with thousands of patients per trial, they've been running trials for myocardial infarction, since the 60s and 70s, worked out that we should be giving aspirin to patients. So really important questions, but we just have not had a great culture, I think, in infectious diseases of doing similar kinds of trials. Now, you could argue that staph aureus bloodstream infections is our myocardial infarction. And so we've been behind the 8 ball on doing these studies. So as an example, with staph aureus bloodstream infections, there's probably been maybe 15, 20 trials over the last 20 years. And the largest of those is 750 patients. And most of those trials sit around the 200 patient mark. And so there's just not been large, well-powered trials. And none of those trials have found a superior treatment to the control. So some of them are non-inferiority trials, some of them are superiority trials, but not one of those has actually found a treatment that's actually improved patient outcomes. So we've come into this spate with our camera 1 and 2 trials and then SNAP. There was a editorial in The Lancet written by some very prominent colleagues, Vance Val and Tom Holland in 2017, that really made a call to say that our community needs to study this disease better and prioritize the right questions and form clinical networks that can get sufficient sample size to answer these questions with sufficient power and precision.
And hence the SNAP trial. I was able to find this New England Journal of Medicine group post made by Paul Sachs. And at the end of the year, he said, what should we be happy for in infectious disease? And number 3 on the list was the SNAP trial in its study of staph. And he says things, and some things about the U.S., that the largest randomized study ever done for staph, SAB, finally brought clarity to treatment questions that have lingered since my fellowship days, his fellowship days, conducted across multiple countries, notably excluding the U.S., which is, he says, a longer story and a longer rant within this. And pointing out this is being done in Australia, and he talks about the various results we'll get to. So it seems that the community is quite pleased and happy and excited about what SNAP's doing at the same time.
Yeah, I mean, ID physicians and microbiologists and people that work in this area love this trial. But it's sometimes easy for us to forget that not everyone in the world is an ID physician and is that interested in it. So sometimes if people, family, friends who are not doctors ask me about what this trial's about, they're like, oh, that doesn't sound very interesting. But ID people love it. And we think it is really important, of course, for given that this is such a common illness and common disease. So yeah, the reception from that part of from ID people and those that are interested in research and clinical treatment in this area has been fantastic. And in fact, it's what's grown the network so quickly in terms of the sites and countries coming on board.
And I think the success to date does relate to the engagement of all our colleagues around the world. So people only recruit to this trial because the questions are clinically relevant to them. As Paul Sachs is saying, these are questions that have been around for 30, 40 years, and we've just not organised ourselves sufficiently to answer them in the way that we should.
Okay, so let's talk a little bit about the trial setup, the structure, the usual clinical trial components of this. So your patients come in with staph infection, blood infection. Do they have to have a resistance to get in the trial? So a lot of this you talk about PSSA, MSSA. What is that? What does that mean?
So we've divided for this trial, and in fact, in reality, it's divided this way as well. Staph aureus into 3 different biological types based on its antibiotic susceptibility. But to get into the SNAP trial, the patient can have any one of these. It's just that they're then categorized according to what antibiotics their organism is sensitive to or susceptible to. The most common kind, which is around 70% of all, is called methicillin-susceptible Staph aureus. So methicillin is an old-fashioned antibiotic no one uses anymore, but it's still called that for historical reasons. And so MSSA is the common kind of Staph aureus that is susceptible to penicillin family antibiotics. Then there's MRSA, methicillin-resistant Staph aureus, that Steve mentioned earlier, which is tends to be resistant to multiple antibiotics, not just to methicillin, and has caused a lot of problems with outbreaks, either in hospital or in the community. So in the USA, for example, there was a strain called USA 300 of MRSA that caused outbreaks in the community, and including among sporting teams, for example. And that causes a lot of skin abscesses and sometimes bloodstream infection. So there's MRSA, which is harder to treat generally. There's MSSA, which is the garden variety one. And then the third kind is PSSA, penicillin-susceptible Staph aureus, and that's like the OG, old-fashioned one, Staph aureus that existed before we started using antibiotics in the world. So pretty much all Staph aureus was penicillin-susceptible at one time, and now between 10 and 20% of it we can still use the old-fashioned penicillin to treat.
Okay, so you refer to these as, at least maybe in the statistical plan, as silos. These are baskets of patients, and every patient fits in exactly one basket. Okay, so these are mutually exclusive categories of it in that. Okay, so within a patient in each of those, you started the trial with 3 domains, you call it. And the 3 domains each have individual questions, and at the same time, you're going to potentially have patients be randomized to each one of these domains. So they could get one from domain A, one from domain B, and one from domain C, one patient in that. So tell me about the first 3 domains. I think the first one was the antibiotic backbone domain, which varies depending on the silo. You described the 3 groups. So what are the questions you're asking in that?
So the questions asked in this backbone domain is really, which is the key antibiotic we should be using to treat this particular patient with this type of Staph aureus? So if it's penicillin susceptible, we're randomizing patients to either stock standard benzoyl penicillin versus an anti-staphylococcal penicillin, which we call flucloxacillin or cloxacillin in some countries. The second silo for MSSA in this backbone domain patients are randomized to flucloxacillin, the Staphylococcus penicillin, or to what we call a cephalosporin called cefazolin or cefazolin, as they say in the US.
No, they say
And then-
cefazolin.
Cefazolin, sorry.
Cefazolin.
And then the third side for MRSA, we're actually looking at standard therapy, which can be an antibiotic, typically vancomycin, in some cases daptomycin, but typically vancomycin. And then the comparator is vancomycin plus cefazolin. So we have this backbone domain. It depends on which silo the patient is in as to which agents they get randomized to. So we're actually answering 3 questions in that domain. And then the second domain is to look in an adjunctive.
Can I go back
Yeah.
to this? Wait, when you're doing this, these are all sort of separate questions, separate patients. There's no statistical borrowing across those 3 for the backbone to give within each silo. So these are somewhat separate, independent questions, statistically,
Correct.
right?
Yeah.
Okay.
Yes.
Okay. So then, sorry, I interrupted domain B.
So domain B is to look at adjunctive therapy. So this is an additional antibiotic to what's already been given in that backbone. And in this case, our first adjunctive antibiotic is an antibiotic called clindamycin, which acts to switch off toxins that have been produced by the Staph aureus bacteria. So it has an antitoxin effect. So we're randomizing patients to either receive clindamycin or no clindamycin for a 5-day period. So that is across all 3 silos. Yeah.
So that could be that could be a valuable thing, even if it's penicillin susceptible, that there's still toxins, and this is almost, as you call it, adjunctive to it. Okay. And there is statistically the analysis of this, is it assumed to have the same efficacy in all 3 of your silos?
It is. We were actually discussing this last time with our statistical team. We are pooling patients from the 3 silos, but we have pre-specified looking at MRSA specifically as well, as to whether there's an interaction between domain A and domain B for that group of patients.
Yeah. Okay. And then the early switch domain C.
So I'll continue. So domain C is looking at whether it's safe to switch to oral antibiotics at an earlier stage than is typically accepted at the moment. So usually for years and years and years, we've been treating staph bloodstream infections with something like 4 to 6 weeks of intravenous antibiotics. So you can imagine patients have to stay in hospital for a long time. They need to have intravenous lines, or what we call, a PICC line, in for a long period of time. We're looking to see whether it's safe and equally efficacious to switch patients to oral antibiotics, meaning that they can potentially get home earlier and have intravenous lines taken out. It's a bit of a dogma that you should continue with the intravenous antibiotics the whole time. So this is kind of challenge to that existing dogma, but potentially has major consequences if we ensure that it's non-inferior to IV for patients and for healthcare.
So how much, sorry, you may have said this, how much earlier is the switch to oral?
So there's actually 2 points at which patients can switch to oral antibiotics. If they have low risk uncomplicated disease, this can happen at day 7 and continue for another 7 days, either IV or oral. If they have more severe, more complicated high risk disease, this then happens at day 14, at which they're deemed to be eligible. If eligible, randomized to then continue IV for another either 2 to 4 weeks, or have all of that as oral therapy.
Okay. So when a patient comes in and they can be randomized among all 3 domains, factorial randomization to that. Does your trial do response adaptive randomization?
We don't at the moment.
Okay.
We've written the core protocol to be future compatible with our AR, but currently all of the domains and all of the comparisons only have 2 interventions. That none of them at the moment have 3 or more interventions. And so partly for that reason, we've not used RAR. We've just used simple 1-to-1 randomization.
Okay. And frequent interim analyses in the trial?
Yeah, well, I don't know what you define as frequent, but after every 500 patients are recruited to the platform and reach their 90-day follow-up, an interim happens. So that's now happened so far. I think we're up to 6,000, around 6,000 patients. So I think the 5,500th patient has had its interim analysis. That's 11.
Yeah. Which I think for most trials that's a big number. More than one or two. And this is a perpetual trial that will continue on. And so every 500 patients, you do an analysis. Do we know any answers? Do we know any answers? Your primary endpoint, we haven't talked about the outcome. What's the primary endpoint of the trial?
It's 90-day mortality. So pretty cut and dried. We debated, should you do a composite? Should you bring it at an earlier time point? Should it be all cause or cause-specific mortality? So those are the kinds of things we discussed. But 90 days, just simple, unbiased, easy to collect, and very hard to argue against that we find a difference in mortality.
Right. And you do, you do have sample sizes to address those questions. I assume a huge issue in other scenarios is your sample sizes are too small for mortality, but the global effort here gives you that incredible opportunity to look at that. You're also enrolling youth and pediatric, it says. So it's adult and peds in the trial.
Yeah, this is one of the kind of innovative aspects of the trial. Is that very few trials in the past, at least in the infectious diseases field, have enrolled both children and adults into the same randomized trial. So most of the questions that are being asked in the trial apply to children who have this illness as well as to adults. So that being whatever age a person is, if they have a Staph aureus bloodstream infection, they can potentially enter the trial. And then there are statistical ways of dealing with children and adults separately, but with borrowing between them.
Yep. And the US FDA just put out a guidance document for Bayesian analyses and all that. It highlights borrowing from adult to peds. And you're enrolling far fewer peds than adults, I assume.
Yes. Yeah. It's something like less than 10% of the patients are pediatric at the moment. So actually 258 children out of nearly 6,000 patients in total. And as you say, the data from the adults can help inform the pediatric statistical testing.
Part of our difficulty is that the primary outcome occurs very, very uncommonly in children. So in children, mortality rates probably sit around 2%, whereas in adults, it's, as described before, it's about 20% usually, and that really increases with age. So once you're 60, 70, 80, much more likely to die than earlier age groups. So that creates some issues with how we do the statistics for this.
Yeah. We should very much point out you have a fantastic statistics team. And you mentioned the meeting of this group. I know from Berry Consultants, Anna McLaughlin was involved in the original designs and modeling and simulation. She's now unblinded and involved in carrying out interim analyses, so thought we would not have her on. And Dr. Julie Marsh heads up your statistics working group, and lots of really cool things going on in the trial. So they do a really awesome job. So we want to make sure we shout out to their efforts.
Yeah, can I just add, I was just reflecting on this last night, that one of my favorite aspects of this trial is learning about the statistical approaches and having discussions and often debates about little statistical questions that arise all the time. And so Steve and I are learning a lot from the statisticians we work with, and really enjoy that, the kind of intellectual challenge of that.
Yeah, it's a beautiful integration of all of that, which is very cool. So you have results in the trial. So you've enrolled almost 6,000 patients, and you've had results in the trial. What have you learned in SNAP?
So we've had 3 what we're calling domain conclusions so far. The first is in the MSSA backbone domain, that we've defined what that is already, but just as a reminder, that was comparing cefazolin or cefazolin, or I like how the English people say it, cefazolin. So comparing cefazolin with flucloxacillin for methicillin-sensitive Staph aureus, so the common Staph aureus. So for that particular organism, flucloxacillin or cloxacillin is we're considering the standard of care and the control arm in the trial, because that's what most guidelines recommend be used. In North America, in fact, cefazolin is more commonly used even before the trial for various reasons, but there are doubts about whether cefazolin is in fact as efficacious as flucloxacillin, and there are theoretical reasons to think it might not be the preferred agent. And so for those reasons, for severe infections, flucloxacillin up until now has been recommended. So it was a non-inferiority design, and we found that cefazolin hit a statistical trigger for the stopping rule for non-inferiority. So the cefazolin, in terms of 90-day mortality, was non-inferior to flucloxacillin. But not only that, there was a higher rate of acute kidney injury, so toxicity from the antibiotics in the flucloxacillin group than in the cefazolin group by a factor of about 1.5 times. So these data have been reported at a conference at ESCMID Global, it's called. It's the sort of largest infectious diseases conference these days that runs every year in Europe. And so the kind of ID community are aware of them. They've not yet been published. They're kind of at advanced review stage at a major journal. So for that first domain conclusion, it already has started to change practice, definitely within SNAP sites, because we have a registry where we measure what's happening outside the trial in patients with staph bacteremia at participating sites, and anecdotally elsewhere as well. But it probably won't be able to really officially change guidelines until it's published in a peer-reviewed journal. But the short version in my mind is flucloxacillin is dead for this indication, and cefazolin is now the preferred agent. Steve, I'll let you talk about the other conclusions.
All right, so let's come back to this a little bit. So now you've got this piece of MSSA where you've got this conclusion. And now what happens to a patient that comes into your site that's MSSA? The backbone is now investigator decision on that, or does the trial deterministically give them cefazolin? What happens now in the trial?
Yeah, the patients, it's an investigator decision now, or clinician decision based. We hope that most will be using cefazolin, but we are not deterministic about that.
Mm-hmm. Meanwhile, that patient, so they would not be randomizing and they would not be contributing to that question anymore, regardless of which arm they get, because they're not randomized, but they still would contribute to domain B and domain C.
Correct. Yes.
Okay. So fantastic. That's the first result. And generally, it sounds like the community has been very receptive, even though it's not out in publication yet, that it's changed care, as you said.
Yeah, we think it has, and that's based on some of that internal registry observational data that Josh mentioned. So patients who are either not eligible or don't consent to the randomized platform, we can consent them to just observational data collection. And so we can see in real time whether there's actually a change in practice outside of the randomized interventions. And in Australia, at least, we have noted that since our results have been publicised, there's been a real drop in the use of flucloxacillin from something like 50% down to about 10%.
Is there a cost difference of the two?
they're minimal. In some areas of the world, so particularly in Europe, actually cefazolin hasn't been registered or isn't on the formulary. So in fact, in the UK, they had to bring in cefazolin onto the formulary for the purpose of the SNAP trial. And so it will take a bit more time, I think, for these results to really impact, because there's greater forces at work. It's not just the individual clinician decision for whether to use cefazolin.
That's fascinating. So maybe even potentially your data could be used to register cefazolin in the EU?
Yeah, we hope that's going to happen. I mean, anecdotally, we've heard in the UK that steps are being taken to get the supply chain working, make the drug available, but we don't know officially what's happened yet.
Very cool. Okay, so result number 2.
So this is the PSA silo for the backbone domain, so our penicillin susceptible Staph aureus. And here we were comparing benzylpenicillin or penicillin versus flucloxacillin. And fluclox is once again considered the standard of care. And part of that relates to the laboratory determination of penicillin susceptibility is not straightforward. And many clinicians and labs actually don't report out on penicillin susceptibility. So this is a little bit of a leap, because we do actually trust what the laboratories say, and we can use that to influence what treatment should be provided. So patients who have been carefully validated to have penicillin susceptible Staph aureus were randomized to penicillin or flucloxacillin, and in this case we had a small patient numbers, so it was about 280 patients, whereas with MSSA it was 1,400 patients. So as a result, we have less power to demonstrate very clear, conclusive findings. But we saw very similar signals, so much more toxicity with flucloxacillin. The rates of acute kidney injury were almost twice as much with flucloxacillin compared to benzylpenicillin. And I think that met a superiority, I guess, probably in terms of the posterior probability of more than 0.99, that it was in fact superior, penicillin was superior to fluclox. And on a mortality counts, so the primary outcome was also better with penicillin compared to flucloxacillin. We didn't quite reach a trigger with that because of the small sample size. And it's worth taking a step back as well. The reason this domain closed was that the toxicity signal for AKI, for acute kidney injury with flucloxacillin, was seen in both the MSSA and the PSSA silos, and therefore the Data Safety Monitoring Committee asked us to stop the trial because of that, principally because of that toxicity signal. Now, the non-inferiority trigger was reached in the cefazolin versus flucloxacillin comparison, partly because of the larger sample size, or with the only 280 patients with PSA, that was not quite achieved, but the very, very similar directional toxicity signals.
So that's interesting. So it strikes me that the MSSA and PSSA are almost similar type groups. They don't have a resistant bacteria at that. So if they're penicillin susceptible, you give them penicillin. If they're not, they're MSSA. Does that mean they're in that bucket? This sort of, and then they might get cefazolin in that case. So does that mean it's valuable to test for penicillin susceptibility?
So I think it is, and what the trial definitively shows, I think, you can trust that test result. So many clinicians would, even if they see it from the lab, would not trust that result. But what we've shown is not only can you trust that result, that in fact penicillin is almost certainly better than flucloxacillin. So these are patients who previously would have just gotten flucloxacillin because the testing was either not done or not trusted.
Fascinating. Okay. So that might even change diagnostic criteria that are used before decisions are made at different ways. Would you ever compare penicillin to cefazolin in the PSSA group or the MSSA? Presumably, if you're in MSSA, you don't test as being sensitive to penicillin.
Yeah. So MSSA partly by definition is penicillin resistant. So that not MS, but in PSSA, yes, you hit the nail on the head there. The cefazolin versus penicillin is an unanswered question, the one that's kind of been brought up after this conclusion. And in theory, yes, we could test that. We've been sort of discussing and debating, should we do that? Is that a good use of resources in the ongoing SNAP platform? And that's not quite resolved yet. So we haven't actually directly compared those two antibiotics, but both of them we know will work well for PSSA.
And just going back to your results, these are for, both of them are specific to adults.
Yes, that's right. Yeah.
Okay. So you haven't had a conclusion in peds yet, is that right?
Correct.
Okay.
And so we actually have reopened that domain for peds. And for peds, we're actually combining the PSSA and MSSA. We're putting them together, and the comparison is now fluvoxetine versus cefazolin for that entire those two patient groups now.
All right, and I didn't miss one. Those are your two conclusions. Was there has not been a third?
There has been has been a third, but we haven't actually reported the detail on that one yet. But what is in, I guess, the public domain is that the platform conclusion has happened, and that the DSMC, the Data Safety Monitoring Committee, has told us to stop enrolment due to having hit a futility trigger. So that was the adjunctive clindamycin domain that we mentioned previously, where people were randomized to get clindamycin or not for 5 days in addition to whatever their backbone antibiotic was. And that after around 4,000 patients were recruited, were randomized, has hit a futility trigger that we're never going to see a difference in 90-day mortality by giving clindamycin. So that's about the amount of detail we know thus far, and we will be presenting those results in detail at the ESCMID conference again this year in April.
Fantastic. So the engine that's generating these results is just awesome. What's the future now? So you've got this platform, and you're interested maybe cefazolin to. And I know you say it 4 different ways. So, and penicillin, maybe there's a comparison in those groups. But what other things are you doing in the platform? What's the future?
So our priority is to try and complete still ongoing domains. So for MRSA backbone, we're up to about 600 or 700 patients there. We'd like to try and get to an answer for that. The early oral switch continues. That's up to about 1,200, 1,300 patients. As far as we know, that hasn't hit triggers yet. And then, because we do have the platform, we'd like to introduce new domains, and there are a number which have been funded now. Some of these are heading more towards, I guess, more experimental therapies, perhaps. So one of them is phage therapy. So phage viruses that infect bacteria, and it can lyse the bacteria. And when the bacteria get lysed, the bacteriophage or the virus gets released and goes to infect the next Staphylococcus aureus. And they're very specific and directed to the bacteria and not any host cells. So phage therapy used to be used in particularly Russia many, many years ago, but with the advent of antibiotics, phage therapy really fell into disuse. But with increasing antimicrobial resistance across the world, there's been renewed interest in use of phage therapy. So we have funding in Australia to introduce a phage cocktail to patients with staph bloodstream infection. So that's one new funded domain that we'll bring on board during the course of 2026. There are also domains to look at adjunctive antiplatelet or anticoagulation agents. So there's some interesting preclinical science suggesting they may be of benefit, but also there are also risks involved in that they could increase risk of bleeding. That we're going to do small, almost really phase 2 trials. So instead of thousands of patients, somewhere in the order of 300 patients into those domains. There's a diagnostic strategies domain as well. So we're looking at PET CT scan. So that's nuclear medicine imaging to look at the hotspots of infection. there's observational data to suggest that doing these PET scans gives you more information that allows you to tailor the duration of your therapy better. So we'd actually like to randomize patients to either PET-CT or no PET-CT scan and determine in a randomized fashion whether this observational data hold up or not.
Diagnostic things like that are strikingly hard. So I compliment you for taking that, because a lot of times the patient is treated exactly the same regardless of what the diagnostic thing says. This has been a plague for cancer for a long time, is that you do the same thing. So it's really hard to show clinical benefit. So that's awesome. It's kind of fantastic because you've shown presumably that penicillin susceptibility is already a good diagnostic thing to add. So that's awesome.
I might just add as a general kind of comment about these new domains Steve's talking about, and he's only got through about half of them so far, is that nearly all of them are not arising from myself or Steve. They're arising from the network that we've built up over time. And that's one of the great things about SNAP, is that there is now a network of hundreds of investigators in the 13, 15 countries. And a lot of them have brought their own ideas to the platform. So, for example, the antiplatelet domain, that's being led by an investigator from Spain, and another one, investigators from Canada, are both doing antiplatelet domains. The bacteriophage is other colleagues from Australia and Netherlands. There's a phosphomycin domain, which is another adjunctive antibiotic from Germany, etc. So a lot of SNAP is, we kind of like planted a tree, and now the tree is like growing and running away from us and getting a life of its own. Yeah. That was a bad metaphor, wasn't it? But you know what I mean.
But what's nice is that the funding isn't all coming from Australia now. Like, each of these investigators has applied to their local funding bodies to support both entry of their patients into the overall SNAP infrastructure, but also to support their specific questions that they would like to answer. Now, whether recruitment to those new domains will be country-specific, or whether actually use the whole of the SNAP infrastructure, I guess we'll wait to see. But the attraction is that once you write a local protocol, we can try and operationalize that such that if there's other countries in the world who are interested in that question, they can also recruit to it and help you get to your answer more quickly.
So we've always described that once you get the adaptive platform trial up and running, like you have, to add these additional questions, to bring in these investigators with these questions, the marginal cost, the marginal lift is so much smaller than had they to design their own trial and go get their own sites. So you're asking more questions than you ever would have, and the marginal cost is less. Is that a fair statement that you've learned, or am I overhyping this?
No, I think that's absolutely true. That is a fair statement, but there are some downsides. There are a lot of added complexity that are hidden, and I guess as an example, we're trying to keep with the principle of a single database for electronic data capture and randomization and eligibility screening. That all happens in a single database, which is fantastic. But we now have a long queue of domains and things to be added to that database, and that takes time, rather than building individual databases would be faster but a lot more expensive.
And you've got all of these have their central cost to the trial, and then there's costs at individual sites. There's governance. So I'm sure you have lots of things you're trying to tackle that goes with that. But that is very cool. So it seems like the adaptive platform trial itself has accomplished a lot of additional goals. Any lesson learned that you'd like to pass off to people? I don't know, regrets is maybe the wrong question, but general lessons learned from jumping into this?
Look, I think we've learned heaps just by doing this. So Josh and I had run maybe 3 or 4 clinical trials, much smaller in scope than this, and it has really just snowballed. So I think we've learned a lot in that process. I think there are some things we would do differently, particularly relating to the database design. I think we've made it a bit too complex with lots of internal logic in the database. That's meant that any adaptations or changes, new domains that we're trying to introduce now, there's a lot of downstream unintended complications because of how we've initially designed things. And I think if I were to go back, I would try and simplify that initial design. It's great for sites because the logic means you get to skip questions that are not relevant to your site or to the domain that you're recruiting to. But some of that, as I say, has created complications down the track now, which costs money and time to try and unpick, and we could have avoided that.
One other difficulty, which this is not something I'd do differently because we don't have control, is that funding cycles, funding agencies, generally government funding agencies, fund trials for a number of years, 3 years, 5 years, for example. And as you mentioned, Scott, we've set this up as a perpetual trial potentially. So as long as funding exists and there are still questions to answer, the trial can keep running. Mm-hmm. However, it's really hard when you're pitching to a government funding agency. You can't say it's perpetual. You have to say you're going to achieve something in 5 years. So we've been lucky enough to be able to get a second 5 years of core funding from the Australian government. And then also in many of the other participating countries, they've had funding from their governments. But it's a lot of work to keep going back and getting a funding again. So it would be really interesting to see. I think just funding agencies have not caught up with this way of doing trials, right, which is still relatively new.
Yeah. That's a whole new podcast of that, which in some ways you're starting to almost do this learning healthcare, where at the 150 sites you have globally, you're learning things. It's implemented probably better and faster. Now you add new questions, and you're doing what we originally said the goal of this was to improve the outcome of patients with staph infection, which was fabulous. So going all the way back to Darwin, I feel like you've improved the outcome of patients in Darwin through these efforts, I imagine.
Yeah,
fantastic.
I think so. That's what you're for. I keep reminding our teams and all the investigators at sites that ultimately this is to improve patient outcomes. We're so in the weeds of the everyday operational aspects, it's easy to lose sight of that bigger picture that because of what we've done and our community has done, I think less patients will die from this disease and less patients will get toxicity from leukoxycillin at this stage, and we'll find out new things as we go.
Yeah. Fantastic. Well, I want to compliment you both on leading this effort, and I know you'd be the first one to say there's huge teams and efforts of lots of people, a global effort, but this is such a really neat example of adaptive platform trial and a global action on this. So congratulations to both of you, and thank you for joining us here.
Thank you, Scott. Thanks for having us on, and I guess thanks for Berry Consultants as well, who have been absolutely instrumental in getting this set up and getting it to work.
Yeah, and again, compliments to Anna for all her work in that. So, and everybody for listening, appreciate you joining us. Until next time, we'll be here in the interim.